My screen. Can you see my screen now?
Yes.
All right. So yeah, thank you. I'm really excited to be here and show you some of the things we are doing in our lab, and I hope to be able to convince you that in plants' roots they still have a number of tricks that we can learn from that we are trying to investigate in our research. I have put here my social media contacts, so if anybody's watching this, please feel free to reach out to me, and if you have any questions or comments to our work, I'm more than happy to discuss this. So we are very fascinated by roots in my group, and one of the most amazing things about roots or plants as such is that they don't really have any shape. In contrast to us, which are kind of boring when it comes to shape, plants they adapt to their environment and they just get the shape they need to accommodate, so to integrate with their environment. And this is really obvious from this picture here, where you can see a root that's adapting to its environment. So in my lab, we have 3 pillars of research. We are particularly interested in root development and in natural conditions or natural light conditions. We are interested in physiology and defense chemistry of roots, and of course also in that connection, root microbe interactions. So we have projects that span these 3 pillars, but overall our goal is to try to understand how root cells respond to the environment and adapt and integrate signals from the environment. But maybe we start a little bit first and say, what is actually a root and what is so interesting about roots as such? Roots are essentially inverted sausages. So if you think about human intestines, we have here inside our intestines, whenever we eat some food, nutrients, they're taken up from inside of our intestines enter into the bloodstream. So you can say you have like an inside out transport, and roots are in fact the opposite. So we have here a root section from our favorite plant, Arabidopsis thaliana. And there, as in any roots, essentially nutrients, they move from the outside of the soil or the rhizosphere into the middle of the root where the vasculature is situated. Roots, they have this very complicated plumbing system inside them. So I'm sure you all know that, there is this vasculature in the roots where we have phloem cells or, fibers that are, evolved to transport sugars and metabolites from the shoot downwards towards zinc tissues. And oppositely, the xylem is transporting nutrients and water from the soil up to the other parts of the root. A very important feature that enables this is that the root has a layer outside of the vasculature called the endodermis. And the endodermis translates literally to the inner skin. So it contains a number of really, really intriguing and interesting factors and abilities that makes all this possible and allow the root to coordinate all these different things. If we take a look at a root here, we go you have the root tip here, and then as the cells differentiate, you start to have this, development of the root, and there are some very interesting aspects happening here that enable these things. So here we have again this cross section of root, and one very particular aspect that enables this transport of nutrients is that in this endodermal cell layer, roots they deposit what's called the Casparian strip, and I'm sure you're all aware what the Casparian strip is. It's a deposition of lignin that's situated in between the cells in the cell walls between 2 endodermal cells. And what it enables is that it blocks transport of diffusion of nutrients in the apoplast here and allow the plant to selectively take up nutrients or water across the membranes of the endodermal cells by expressing distinct transporters or channels in the endodermis. It's in fact similar to what we do in our intestines also, where this ability is facilitated by something called the tight junction. So this is very an interesting analogy between plants and mammals, so to say. I like to think of this as if you have a window, right? You can see here small windows, and the light you can consider the water or the nutrients. It can go through the cells or the glass, which is here the membranes, but in between the cells you see it's blocked, so no light can pass through the cells. So it gets filtered. You can maybe get rid of UV light or whatever. But interestingly, so in contrast to intestines, roots are actually a bit more advanced. They have another system. So if we move here a little bit further up the root, we get something called suberization. And what suberization is essentially that now the cell or the membranes can be covered in another layer called suberin. And this creates another barrier. This is essentially a polymer of waxes and aromatic polyesters that cover this. And you can compare this to closing the shutters. No, so you all know suberin because it's essentially cork, and you know how cork is extremely good at keeping things in bottles, for example. So it's very hydrophobic. So there's not a lot that can diffuse out through this. A very interesting thing that happens in this zone here where this suberin is deposited across the endodermal cells is that some cells are not depositing the suberin, and these are called passage cells. So these are simply defined as unsuberized exo endodermal cells that are adjacent to the xylem pole in an otherwise suberized area. These were discovered already more than 100 years ago to exist across multiple species of plants. But in fact, we don't really have any function or know what they do so far. So you can see here is a cross-section of the root. You see the suberized cells here, and you see one unsuberized cell in the xylem pole. We actually have markers so we can see these cells live. You can see here is an endodermal layer where you see a yellow is a marker that is highlighting suberized cells, and below you see the xylem pole in red. So you can see this window here through the cell. If we follow the analogy from before, it corresponds to certain tiles or certain windows being open in an otherwise closed area. So we can say, if we have this passage cell, we like to call it the open door policy. So you can say you have this area here where you are not allowed to pass. It blocks everything. It ensures nothing can come in. And then our hypothesis that, maybe you have this hole here where, okay, you can allow selective entrance or communication through these passage cells which provide entry into the xylem. So this is our idea, and this is also essentially why these things are called passage cells from the beginning, but we have no evidence for this throughout history so far. There is one problem studying passage cells, though, and that is that old cells, and in particular these superizing and Casparian strip containing cells, they are tough, so it's very hard to study them. And another problem within developmental biology is that we have what I like to call the comfort zone, which is the meristem and some young cells. These are easy to study. You can see that, and very often studies are focused in this area of the root. And then we have what I call the developmental outside the comfort zone, which are these old cells because they're quite tricky to study. Now we have been chasing these passage cells for a while, so we had tried many different things. We have done some single cell analysis where we've been able to protoplast some of the young cells here, but one of the problems we see is that we never really see any of the markers that correlate to the suberization come up in single cell analysis, and that's of course because there is an inherent problem with the single cell analysis is that you need to have cells that are protoplastable that you can isolate. Otherwise, it's tricky. And this is tricky when the cell surface is covered in suberin, for example. So we collaborate with Bert de Rübel from the VIB in Ghent, and we have other methods. For example, we are using translating ribosome affinity purification, where we can express a ribosome tags specifically in these old cells, and we can grind up the root and extract information that way. But recently, we also switched to single nuclear sequencing, which is also extremely powerful. What you see here is a recent study where we isolated about 300,000 nuclei from the cell, and we were able to actually do a pseudo-time trajectory of the entire endodermal development. So we have down here meristematic young cells, and we have a developmental gradient onto old cells. We can see actually genes related to the suberin synthesis are coming up in these later parts of the cell, and we actually have some beginning evidence that maybe there are some other trajectories in this that could potentially be passage cells. So we are onto this, and we're really excited about starting to learn what the passage cells are doing. But one thing I'm particularly proud of and particularly interested in showing you is how we study passage cells in vivo. How can we do this in actually a live root? And one of the biggest problems when you study roots under a normal microscope, for example, is that in a microscope, you always put a slide, and that means that you put your plant or your root horizontal, right? So this is really not good for plants because roots, they like to grow downwards. They will always try to grow downwards. They will always bend towards gravity. So within a few hours, you have a root that's actually responding and stressed in this situation. So the way we solved this was that we flipped our microscope 90 degrees. So we mounted it on a vertical plate like this. And now we can actually allow the roots to grow downwards, and we can study them as they move along. But then again, a slide here, growing a root on a slide is also not particularly optimal. So we developed some growth chambers where we can seal off light from the top, and we can also grow the roots inside artificial transparent soil matrices that are customizable. We can also control the light. We can control the nutrient composition locally even, so we can adapt and we can study how different parts of the root responds to different abiotic or biotic conditions. We can also include, for example, microbes in this situation. So this is very nice. You can see here, for example, we were able to image a seed as it germinates and as the root keeps growing downward. So we can grow roots for this, essentially as long as it takes. We can image. I think our record is currently, 14 days of continuous imaging where we can get images. And that's, of course, huge data sets that contain all sorts of other problems. But so just to show you an example of what we can do with the passage cell. So if we have the root here, where we have the suberization and the passage cell establishment in the old parts, if we start to image this zone right before the suberization is happening, here you can see a marker for a gene that's involved in suberin biosynthesis. And you can slowly see how the suberization is onset here. And then we get this pattern. And you can see there are some holes here in the suberin, and this is actually the passage cell. So this is what they look like live. You see there is quite a number of them, and this number appears to vary, and it varies depending on different conditions. So you can study how different conditions are affecting the passage cells. So we have this situation, and in collaboration with Pau Formosa Jordan from our local institute, we are trying to see, can we take this to the next step and study single cell responses in vivo in real time. And the way we do this is that we constructed about 500 different reporter lines for different genes, which are mostly transcriptional reporter lines. And then we have an imaging pipeline where we image the roots over time. We can then, we are working on the 4D segmentation of these to get this to work, to study responses. And that way we can map and see how each cell is, how much of a signal we detect over time. So here you see, for example, a marker. So you see another problem here, for example, if you use transcriptional nuclear markers, is that the nuclei are extremely, mobile. So you also have to do tracking of these signals, right? So, but essentially we can here for one cell, we can see in yellow, you see the suberin signal, this goes up over time, and you see here's expression of a marker in a passage cell, you see how the signal goes down over time. Here we imaged for about 108 hours in this particular root. So we can now gain insight in single cell behavior in a root that's actually living in an environment that's customizable. So just to recap this, we have this image platform that allows us to study the single cell behavior in living roots across, a number of different conditions we can customize in our setup. We have this toolbox, and we have about 500 transcriptional reporter lines. These include a lot of nutrient transporters, defense genes, and a lot more different things we picked up to be passage cell associated. We have actually recently identified the first known repressor of passage cells. I actually just submitted the proof, so hopefully this paper should be online very soon. But of course, you're all thinking, okay, so we can study passage cells, but what do they actually do? What is the role of passage cells? And this is really a question that intrigues us a lot. So we have, of course, Arabidopsis, which is really a very nice plant, especially for microscopy. It's essentially nice because it's a small plant, but it's also very limited when it comes to particularly biotic associations. And this is where we're using a different model. And this is actually a legume. It's called Lotus japonicus. And Lotus has this fantastic ability to form these nodules on their roots, which can host rhizobia. And rhizobia can take up nitrogen from the environment and provide ammonium or amino acids for the plant in exchange for carbohydrates from the plant. So this is really an amazing ability from legumes to do this and allow them to grow on very low nitrogen soil. And this has been pursued by numerous of consortia and different things to try to understand and integrate this and figure out how we can use this in agricultural settings. Very recently, we actually made a discovery, and we connected the ability to form these Casparian strips in roots with the ability to form functional nodules. So we identified a mutant that's unable to make Casparian strips, and it also has a strong nodulation phenotype. So we have established a toolbox to study the role of barriers in this particular plant. But I'm not going to talk about the Casparian strip. I want to show you what's very interesting when it comes to passage cells. So what you see here is now a nodule primordia developing here on the root, and you see at the same time a staining method we developed a couple of years ago, where we can look at suberin and lignin at the same time here inside the root, and you see in yellow are the suberized endodermal cells, and you see here below you see xylem pole, and you see the stars are highlighting passage cells. So there's actually a very intriguing connection between nodule primordia and there's passage cells happening adjacent to that. It's a little bit easier if we look at a cross section here. So here you see a cross section of the root. We can see the xylem poles are actually here. So in here we have the xylem that has 4 poles here. We have passage cells situated here. And then what we think is happening here is that we have a nodule primordia that it's always developing in the xylem pole. So this has actually been established already in literature that there's a xylem pole association to nodules. But what we're thinking is that there's likely a bidirectional communication here. I mean, there needs to be a developmental connection between establishment of connection to the vasculature beneath here. So I think it's very reasonable to think there is a connection that needs to be established between this. So this is one of the questions we're pursuing in terms of what are the functions of passage cells. To do this, we had to come up. Lotus is not so easy as Arabidopsis to deal with genetically, so we had to come up with a different methods to do this. So together with Ton Bisseling from Wageningen University in the Netherlands, we did a laser dissection study where we took sections of the root and we were able to dissect out regions in the xylem of phloem poles. It looks something like this in real life. And then we could take these different sections together with Novogene. We sequenced this and looked for genes that are enriched in the phloem pole region, which we assume associated with the suberization. These are, of course, known from Arabidopsis. But then what was particularly interesting was to identify cells in the xylem pole and see if these were related to passage cells. So overall, using this method, and we went quite stringent actually here on our threshold. So we identified 118 genes that were specifically enriched in the xylem pole region, and we took the top 20 candidates of these. And then because Lotus is not easy to transform, we had to find another way to visualize these genes. So Swati, my PhD student, she developed in Lotus a method of fish where she could look for the messenger RNA themselves inside Lotus. This is a fascinating technique. And you can buy these probes from molecular instruments. These are really, really good. They're very specific and very powerful tool to look at this. So you can do this whole mount in Lotus. So here you see expression of an endocortex enriched gene. This is Casp1. So Casp1 is actually involved in Casparian strip formation. And you can see we get a very nice specific signal here, particularly in the endodermis. And one of our candidates, actually a number of our candidates show very interesting patterns like this, where individual cells inside the endodermis are highlighting. So this is, of course, very, very exciting. And we think we have found some genes that are specifically expressed in the passage cells. So now we, of course, mutating these genes and trying to see, can we find some phenotypes on these plants, and also doing numerous of different experiments to investigate what are these doing in Lotus, and do we actually now have a hold on genes that are really particularly expressed in passage cells. So we think that if we have a very simplistic root as Arabidopsis, right, and we have a very specialized root like Lotus that can form these fascinating structures, maybe here we have this connection where these nodules are connecting through the passage cells and allowing communication with the beneficial bacteria. Whereas maybe in something like Arabidopsis, you have a similar situation, where you're kind of communicating with beneficial bacteria that are sitting on the surface of the roots. So where you could say in a simplistic setting, maybe you have like a rudimentary microbial communication, where you have here a situation with a highly specialized communication that has led to evolvement of communication inside these specialized structures. So this is one of the hypotheses we're currently trying to gain more insights into. Of course, we also try to put the Lotus system into our growth setup. And here you can see we can actually grow Lotus plants. We can get them to nodulate inside our growth chambers on the microscope. And we have been able to image this. So this is unfortunately not yet fluorescent, but this was technically extremely challenging. I think this was 15 days of continuous imaging of a root, and you can see actually up here that a nodule is forming on the root. So it's very technically challenging to do that, but we are making steps to image these processes in the near future. So take-home messages from my talk is that passage cells are specialized root cells that likely contain overlooked functions. We are now actually starting to gain tools that have the sufficient resolution to investigate this. So beforehand, we haven't really had the resolution to look at this. So with the new tools we're developing and other things that are coming, now we can actually really start to gain single cell resolution in vivo on some of these things. And also, of course, one thing we are very interested in is expanding this into non-model species, and maybe this can give us new models or new ideas for how root communication is actually happening and how individual cells inside the root can play important roles in this. This is, of course, extremely important because if we want to make more resilient crops that can maybe talk better with their microbiomes or determine which microbes they want to recruit, this is one of the biggest unknowns when it comes to plant microbe interactions is how are the good guys distinguished from the bad guys, right? This is some of the questions that we might be able to dig further into. With this, I would like to thank a number of collaborations and help that have been incredibly helpful throughout my start of my career to establish my group and also give input to what I've shown you today. Of course, I would also like to thank my amazing group for all their effort and all their amazing experiments and their dedication. In particular, I would like to thank Defang, who is my Lotus master, who really has done most of the things I've shown you, together with Basti Samwald, who has done also some of the long-term imaging I've shown you, and Swati, who is also Lotus expert and has done all of the fish analysis. Of course, a number of other people have been involved in this that I showed some data from. So particularly Eric, Noah, Jungkum, and Juan, who have all contributed to this also. I would like to thank all of our funding. And of course, I would also like to thank you for your attention and welcome you to contact me if you have any questions or comments to our work. Thank you.
Thank you, Dr. Anderson. Very nice talk. Now it's the time for our Q&A segment with the committee members. So for all the committee members in the meeting room, if you have any questions with Dr. Anderson, please feel free to turn on your microphone. Dr. Ralph, please.
Yeah, thanks for a great talk. According to the images that you showed, the spacing of these cells, these passage cells, looks quite regular. And I was wondering how strictly defined is the patterning of these passage cells within the root? And do you have any ideas how the patterning is regulated and what are the factors that influence it?
Yeah, very good question. Thank you, Ralph. This is intriguing. So it seems to be that it happens in patterns within patterns. So we're actually trying to model this. We're trying to measure these differences and see are there actually some kind of underlying pattern to this. And it seems to be not only dependent on the position in the root where you are, but also the age of the root matters. And also, sometimes you never get passage cells in the phloem pole, but sometimes you get them in the cell that's next to the xylem pole. And we think there's multiple different mechanisms that are influencing this. We did actually a randomization test. We saw, is there truly different from random, in terms of stochastic occurrence, and we couldn't really determine anything. I know you cannot really say is anything really random, but we think there is a number of different developmental programs that influences this decision. So short answer is it looks regular, but it has many different components to it that we still don't fully understand.
So the modeling that you've done so far, is that compatible with the existence of like diffusible substances that establish gradients, like patterning occurs in other systems that undergo cell differentiation,
Exactly.
like the heterocyst system in cyanobacteria being the simplest one, probably.
Exactly. It is the best fit we have is with a diffusion model, and we think the signal that is involved in this is ABA, and we think that it's ABA diffusion, but we think there's a pre-programming that happens in the very young meristem, where specifically the xylem pole associated cells are influenced and have a different threshold towards presence of of ABA. So we think the moment that an ABA wavefront, so to say, is moving down to the root, the xylem pole cells are more resistant to that wavefront. So only certain of the cells might go above the threshold and then suberize, but the imprinting that happened in the very young endodermis or very young meristem might prevent this from superizing in cells that are destined to become passive cells. So this is kind of like a spatial temporal model, you would say.
Great, thank you.
Thanks.
Chris, please.
Hello, Tony. Thank you very much for this nice talk. I was wondering whether
Thanks,
when you have conditions such as drought, which leads to enhanced suberization, you ultimately also block these passage cells, or are they sort of constantly protected from suberization?
Yeah, this is also a very good question. Thank you. It's a very interesting aspect, in fact, because suberin is not a final decision, so to say. So we have evidence that suberin is quite dynamic, in fact. So you're absolutely right that upon drought, cells superize, passive cells also close upon drought. I mean, if you add ABA just to the root, you see all the passive cells, boom, they will close immediately. So, but then again, you remove ABA and you leave them for a while, then you can actually start to see passive cells again. It's very tricky to measure because we have transcription markers for suberin, and we have a way to measure suberin itself, but that we cannot do live. So we have to rely on the transcription markers. So it's a correlation at best, but we can absolutely see if we do a drought or ABA, you see no passive cells. If you wait a couple of days, do on the same stressed root, you see passive cells again. So there is absolutely a dynamics to this. So I think the roots really need them, and they want to ensure that they have them open when they need them.
But given that the root grows, are you sure it's the same cell which was suberized and then desuberized, if I can call it that, or the new cells, once you change the media or the conditions, then keep or maintain these passage cells?
You can of course say that, but you can also say, look, you can look in a zone that no longer grows, right? I mean, there is a suberized zone in, let's say, a 5-day-old seedling, right? And if you then look later in that suberized zone, right, these cells were suberized from the beginning. And in fact, a very interesting aspect we did was that we made a marker that inducibly can repress the phenylpropanoid pathway in the endodermis. And that was really interesting, because once we then measured the suberization before the induction and during the induction, we could see that the suberization pattern or the fully suberized pattern became shorter and shorter and much quicker than the root was growing. So there is really evidence that this is actively being degraded. We imagine it's probably a weight between synthesis and degradation, and the outcome is like a homeostasis. And then it's very nice because if the plant wants to very quickly suberize, it switches that balance a little bit towards superization, and vice versa towards degradation if it wants to have more passive cells and more open suberin.
Thank you very much.
Thank you.
Thank you very much again, Dr. Anderson. I think it's the time. Thank you. I think it's the time to move on to our next finalist, Dr. Zhen Yan from Westlake University. Today, she will share her story about the gateway to chloroplast protein import. Dr. Yan, please.
Okay, thanks for the kind introduction. I will first share my screen. Okay, can you see my screen?
Not yet.
Oh, I will try again.
Oh, yes, I can see it.
Okay.
So, yeah,
here?
That is not no. Yes, it's a PowerPoint now.
Okay. So Okay, this is Jun. Hello, everyone. This is Jun from Westlake University, and it's a great honor to share our work at this 2025 Rising Stars in Plant Sciences, our presentation. First, I want to briefly introduce myself. I got my PhD, my bachelor degree from Nanjing University and PhD from Tsinghua University. Then I worked as a postdoc at Tsinghua University and Princeton University successively. And I joined Westlake University since 2019. So during my PhD and postdoc period, I mainly focus on the magnetic and the structural investigation of KN channels in excitation, contraction coupling pathway during muscle contraction. And since I set up my independent lab at Westlake University, I began a new direction, which is to focus on the process of chloroplast protein translocation. I made this transition from ion transport to protein transport for several reasons. First, protein translocation and targeting is very fundamental, but in chloroplast, the understanding is likely far behind. And also, since I set up my own independent lab, I want to step out of my comfort zone and establish my own academic identity. Okay, so today I will only talk about the work at Westlake University. Now, please allow me to take you through our exploration of the unlocking the gateway to chloroplast protein import. So, when talking about the most important chemical reaction on Earth, the first answer that comes to your mind is probably photosynthesis. So, the chloroplast is the primary site for photosynthesis. It serves as the core hub of biosphere energy conversion, which drives over 99% of Earth's primary productivity. And for chloroplast, the function of chloroplast is far beyond photosynthesis and also deeply involved in multiple essential physiological activities. And also, chloroplast-based engineering holds a significant application value. But for all of these activities and applications, they all rely on the proteins within the chloroplast. So in chloroplast, there are about 3,000 proteins, and 95% of them are encoded by the nucleus and then synthesized in the cytosol as preprotein with an N-terminal transit peptide, which will direct them to get into the chloroplast through TOC and TIC. TOC and TIC are the translocon at the outer chloroplast membrane and the inner chloroplast membrane. And for the chloroplast protein translocation is an old field with a history spanning 3 to 4 decades. Nobel laureate Gunther Blaubel pioneered this field and together with his postdocs identified the core components of the TOC system. However, research on TIC has been controversial. Despite the persistent efforts of scientists worldwide for decades, there still no consensus has been reached. So here's a brief summary of the situation, the consensus TOC and controversial TIC. For the TOC, it consists of 3 components, TOC75, which forms the conducting pore for the substrate preprotein, and it also contains a member from TOC34 family and a member from TOC159 family. The 3 of them constitute this translocon as the outer envelope membrane. But for TIC, the composition which protein constitute this translocon has sparks a lot of debates. There are 2 major models proposed with quite different composition, different components, but both models agree that TIC20 is a key component. So although this field has a long study history, but still a lot of fundamental questions remains to be solved. For example, what's the exact components of this machinery and how do TOC and TIC form a supercomplex to translocate the preprotein to get across the 2 chloroplast envelope membranes. So in the past decades, numerous strategies has been employed, such as cross-linking, antibody co-IP, and genetic mutations, but still no clear answer. So we would like to take an ultimate approach, which is the endogenous purification and structure determination, to directly answer these questions. So our idea, first, to isolate the endogenous complex, our idea is to add a tag to the consensus component to pull down the native whole complex, to rule out the potential artifact caused by the tag, and to rule out the debates between different models. We choose two consensus components to add tag for mutual verification. One is the consensus component from the top complex, and the other is the only consensus component from the tick translocon, so that in total 3 strings were made for cross-validation. And then we purified the top tick supercomplex from the 3 strings respectively. And the purification result shows very identical both on the gel filtration and assets page, which indicate that we isolated the genuine the endogenous top tick supercomplex. To cut the long story short, after a lot of trials, we finally solved the structure of this top tick supercomplex. As you can see here, it is very huge, spans from the outer membrane to the inner membrane. And according to this structure, a total of 14 components was identified, and also a few of them are previously uncharacterized and are identified by this structure. And here, this is the top part, and to our surprise, that previously thinks that the top 75 forms the conducting pore for the substrate, but actually in this structure, the top 75 and top 120 forms a hybrid beta-baryon pore, which assembles the bacteria assembly machinery, BAM-A. This indicates an evolution adaptation from the cyanobacteria. Okay, so for the tick part, they consist of the all the components from the second model, but not the first model. So here, according to the structural analysis and based on the endogenous purification, we finally answered the decades-long composition question of the tick translocon. So according to structural analysis, we carefully and also confirmed by the cross-link mass spec, we proposed the working model for the top tick translocon. The pre-protein first enters the hybrid beta-baryon and proceed along the hydrophilic cleft on the intermembrane space scaffold, and then reach the TIC20 and YMG enclosed negatively charged cavity, and finally get into the chloroplast. Okay, now let's come back to this slide. Now we have characterized the translocon, and you may have found another question mark here. So actually for the protein protein translocation in chloroplast, it is a process that requires energy, which comes from the hydrolysis of ATP. So ATP-driven impulse motor is essential for this process. But what proteins constitute this motor? It sparks even more debates than the tick. Two models has been proposed. Each entirely contains entirely different components. The first is the chaperone model, and the other is the YSF2 FTSHI complex. So between the two models, there's even no one consensus component shared between them. So our previously successful strategy to focus on the consensus component and to isolate the endogenous complex will not work this time. Then how to identify the chloroplast impulse motor? So to qualify as a chloroplast import motor, there are two golden standards to be met. First, it has to interact with the pre-protein at very early stage. And second, it has to physically interact with the TIC translocon for their mechanical cooperation. And to match with these two criteria, our goal is to capture the ultra complex, which constitutes by the translocon, motor, and pre-protein. And then through structural investigation, we can identify what is the import motor. And then first, we set up a chloroplast import assay. We isolated intact chloroplast from pea leaves and prepared a tagged pre-protein. And then we began the reaction. And in this gel, the antibody recognized the tag on the pre-protein. And the hair band represents the pre-protein, and the lower band represents the motul form since the signal peptide is removed. As you can see, as time went by, the pre-protein gradually translocated into the chloroplast and processed into a mature form. And then we stopped the reaction at a intermediate time point. And then we purified the whole complex through the tag on the pre-protein. And here shows the mass spec list of our purified sample. At the top candidates, you can find a lot of components which belongs to the top TIC, and also we found some components belongs to the YSF2 FTSHI complex. This result indicates that the YSF2 FTSHI complex may function as this import motor, but it is still premature to draw this conclusion, since it is known that co-IP often introduce contaminants which only a non-specific binding. So we still need structural investigation. Okay, then we collect a lot of data, but unfortunately, due to the intrinsic dynamic, we failed to achieve a high-resolution cryo-EM map for this ultra complex. We only obtained, despite of our best effort, we only obtained a moderate resolution map with a resolution about 11 angstrom. At this resolution, we cannot identify the proteins according to this density map. Then we turn to a divide and conquer strategy. So that is to solve the high-resolution structures of each complex and then phase them back to this ultra-complex map. Then we start with a relatively easier part. This part looks like the toptic structure we previously solved, but this ultra complex is from P, the land plant, but the structure is from Chlamydomonas, is green algae. So direct structural alignment will show less accuracy. Then to get accurate comparison, we go back to land plant to solve the structure of the translocon. And we, based on our previous experience with the Chlamydomonas system, we choose to add a tag on the TIC20 and to generate the transgenic line and isolated the endogenous translocon sample and solve this structure. And the overall architecture and components are conserved with that in Chlamydomonas, but the local assembly differs. And then, according to the newly solved structure, we confirm that this part is the toptic. And then, what's the other part? The motor part, what protein constitutes this part? Then we go back to the purification result of this ultra complex. Since the components of the YSF2 FTSHI complex has a very high rank, so we want to solve the structure of this complex and compare with this ultra complex map. Then we purified the native endogenous YSF2 FTSHI complex and then solved its structure. Here, this structure contains all the 7 previously reported components, and also according to the structure, we identified 4 new components. At the stroma site, it forms a heterohexameric AAA ATPase motor module, and the 4 new components position this motor module at a tilting angle of this motor relative to the inner envelope membrane. So this tilting angle may have some physiological functional implication. Now, with all the structures at hand, the most exciting moment came. The 3 maps are shown in 2 views. As you can see here, the YSF2 complex is very like this part of the ultra complex, and this TIC complex is very like this part of the ultra complex. So when we align the 2 high resolution structure with the ultra complex map, they matched very, very well. These findings directly and powerfully confirms that the YSF2 FTSHI complex is the import motor of the chloroplast protein import system. A long-standing mystery has finally been solved. And now, based on this density map of the ultra complex, I want to revisit a previously mentioned question. What's the physiological significance of this tilting angle between the motor module and the inner envelope membrane? As you can see here, because of the tilting angle, it enables the close contact between the TIC and the motor module, which will facilitate the handover of the preprotein from the TIC to the motor. And according to all the above data, we propose a working model for the chloroplast protein import system. First, the protein enters the top TIC and will be recruited by the pour loop of this motor module. And then the ATP hydrolysis will generate the pulling force to propel the preprotein to get out of TIC. And then the preprotein will be released to the stroma, and the whole machinery will be ready for a new cycle for the preprotein translocation. Okay, the story, however, does not conclude here. While the translocon and the motor in land plants has been extensively characterized, so the structure and functional organization of the motor complex in green algae has been remains elusive. So whether in the green algae also have motors that conserved to the land plant. So then we go back to the starting point, the Chlamydomonas, to study this motor, to reveal the evolutionary conservation and diversity of the protein import system. Okay, so after a lot of trials, we managed to solve the structure of this YSF2 FTSHI complex in Chlamydomonas. Based on the structure, we identified 19 subunits, which is much more than that in the land plants. And among the 19 components, 5 of them constitute the heterohexameric motor module, and there are 14 accessory components associated with this motor module. And also, the motor module shows a fixed tilting angle relatively to the inner membrane, which will facilitate the preprotein handover, and it is conserved with the motor in the land plant. Okay, in accordance with the structural observation, the purified motor complex exhibited robust ATPase activity. Notably, this activity was increased when incubated with the preprotein. This may suggest that the preproteins interact with the motor complex as translocating substrates, thereby enhance the hydrolysis of ATP. So now from a biochemical view, we once again proved that the YSF2 FTSHI complex functions as an import motor. To gain a more comprehensive understanding of the evolution of the YCF2 complex, we further analyzed the presence of these components across different species. These components are mainly found in the green lineage, but largely absent in the red algae. So this motor complex may formed after the divergence between the red algae and the green lineage, these two branches. And then within the green lineage, the two structures we saw in the Chlamydomonas and Arabidopsis are conserved within the green algae and land branch, these two branches respectively. And between them, the motor module, the core are conserved, but for the accessory subunits, they shows great diversity. So this may indicate these accessory components may gradually incorporated to this motor after the divergence between the green algae and land plant. And it is very interesting that the Poaceae species here, it lost all the key components of these translocation machineries. So it is known that the Poaceae species undergo dramatic genome rearrangement during evolution. So this may lead to the loss of these components. And these Poaceae species may have evolved alternative protein import systems, which requires future further investigation. Okay, to brief summarize, we identified the composition of tick, elucidated the molecular mechanism of the toxic translocon, and characterized the YCF2 FTSHI complex as the import motor. And also we reviewed the evolution diversity across species. And through structural investigation, we provide a clear answer to the decades-long controversial models regarding the translocation machineries and drawing a whole picture of the chloroplast protein import process. So last but not least, I would like to thank all my lab members, especially Zeyu, Lianghe, and Yuxin for their hard working, and also deeply thank my collaborators for their strong support. Furthermore, I'm extremely thankful to the Westlake University and Westlake Laboratory, and also the NSFC for the funding support. And thanks for your attention.
Thank you, Dr. Yan. For other committee members in the meeting room, if you have any questions with Dr. Yan, please feel free to turn on your microphone. Ralph, please.
Yeah, thank you very much for a great talk. So as you said, the grasses have lost the YCF2, but they've also lost the YCF1, right? Which, according to the model that you now provide, which is of course quite compelling, is a key component of the tick complex. So have you tried to use your tagging purification approach in rice or any other monocot? And what's your current thinking about the tick composition in the grasses?
Yeah, thanks for your question. It is the key question, that direction we are ongoing. But unfortunately, we tried the tagging strategy, which we are good at, and also the input assay in the Poaceae species, like the rice, wheat, and maize. We tried these three species. But unfortunately, by now, we haven't got a reasonable result to identify whether a stable complex are involved. So we through tagging, we cannot isolate a well organized complex. And also for the import assay, by now we still not success on this part. but it is the most important future direction for us.
And what's your general thinking on the evolutionary dynamics and flexibility of the whole apparatus? I mean, as you showed already between kilometer monas and a high appliance, there are some differences, especially in the auxiliary components of the machinery. And then, as we just discussed, the YCF1, YCF2 losses in some lineages. So why is that? Why is the whole system not as conserved as other large molecular machineries like the mitochondrial protein import or the ribosome? What makes the chloroplast protein import machinery so special and creates the need for this evolutionary flexibility?
So it is an interesting question. The short answer is I don't know, but but I think so. They are not strictly conserved, but as you can see, the core components they are for motors they are conserved, and also for the TIC translocon, the TIC20, a key component, they can also be found across almost all the species. But the other components like YCF1 and YCF2, I think so. We know that these two proteins are quite large, and maybe during evolution, there might be some truncations and different construct of these two genes, so that now we cannot identify them by simple sequence alignment or something else. So it is interesting, but now I cannot have a clear answer to this question.
Okay, thank you.
Thomas, please.
Yeah, so really great work, Sen. I'm sure I will show the super complex in my next student lecture. So I have a more mechanistic question. So this AAA ATPase complex, which is necessary to pull the protein through the TOC-TIC complex. So how does it dynamically function so that really a protein is successively pulled? And so can you say anything about the dynamics? Is a complex that somehow goes like that? Or so do you have any ideas how it pulls the protein in the end?
Okay, so during the structural investigation, we have different classes actually. So for this motor module, there are 6 components, and almost every one of them, and extremely some of them shows the high dynamic, and the density is very weak due to their intrinsic dynamic. So during translocation, this motor module may show dynamic modes. And also, there is very interesting that I didn't analyze in detail that the 6 components here, only 5 of them has the ability to bind and hydrolysis ATP, but for YCF2, it lost the consensus motif, and it don't have the ability to bind or hydrolyze ATP. So we also discussed the working mode for this motor. At least it won't adopt the hand-over-hand mode because of the asymmetric of these 6 components. But for the exact the path of the pre-protein go through these translocon, it is only a proposed model. Unfortunately, we didn't capture a structure with a substrate bond. So now we cannot have a conclusion that where is the exact translocation path, but we are still ongoing.
Yeah,
Okay.
thank you.
Maybe we can have the last question. Dr. Cui, please.
Yanjin, very nice talk about the TikTok and also protein import into the chloroplast. I'm wondering how chloroplast maintain protein homeostasis or even export some proteins. Is the TikTok involved in these processes, or how the TikTok activities dynamically regulated?
Okay, thanks for your question. That yeah, for the homeostasis of the protein within the chloroplast. So for by now, according to the reference, all the reference now, the function of this tactic is only for the protein import. For export, so far there's no report for the tactic, but there's some degradation pathways. For example, in recent years, Paul Jarvis proposed the chloride system, which is correspond to the ERAT in ER, that will degrade some unwanted proteins, and also it will regulate the TOC complex, the component, so that to regulate the function of this translocon. So according to this import and also the degradation, and together maybe with some other uncharacterized system together to maintain this homeostasis of the protein within the chloroplast.
Okay, thank you.
Thank you very much. Thank you very much again, Dr. Yan.
Okay, thanks. I will stop my sharing.
Okay, let's move on to our next finalist, Dr. Jizhou Wang from Beijing University. Today, he will share his story about plant phytochrome signaling. Dr. Wang, please.
Yeah, thank you. Okay. Hello, everyone. It's my honor to introduce my research work to you. We know plants adapt to the environment by interactions with diverse abiotic factors such as light and temperature, and biotic factors such as pathogens and microbes. I have studied plant immune receptors during my graduate study and postdoc. We know two types of immune receptors exist in plant cell: the cell surface receptor and the intracellular receptor. But how they activate immune signaling remains unclear. During my graduate study, I identified SERG protein as the co-receptor of PSKR-mediated growth defense signaling. How did I find it? I first solved the structures of PSKR in free and PSK bond forms. Then we found PSK binding induces the formation of a small island domain called PSKRID, the light red part. This suggests other unknown components might bind the newly formed PSKRID. Then our biochemical, genetic, and structural data all support SERG members function as co-receptors of the PSK signal transduction. Then in my postdoc, I turned to investigate the intracellular immune receptors and discovered the plant resistoosome, and further suggested its function as an ion channel. Again, I first solved the structures of the ZR1 ACAS1 immune receptor complex in both resting and UMP modified PBL2 bond forms. Then we found PBL2 UMP, the green part, binding triggers the conformational changes of the nucleotide binding domain of ZR1, called ZR1 MBD, the light red part. Then the EDP releases from ZR1. Our biochemical and structural data further reveal that DETP or ATP promotes the formation of a wheel-like ZR1 pentamer, which we called resistoosome. Amazingly, the CC domains at the antinomous of ZR1 resistosome form a channel-like structure. Is the pink part, as its outer surface consists of hydrophobic residues such as leucines for membrane binding, and its inner surface consists of negative charged residues such as glutamate 11 and 18, probably for calcium ion permeability. Importantly, mutations on either leucine or glutamate residues could abolish the function of ZR1 resistosome. As a recognition of our work, the 2023 International Society for Molecular Plant Microbe Interactions Congress took our resistosome as a part of their logo. The decade scientific training made me realize that bringing new approaches such as structural biology to an old problem in plant science can lead to major breakthroughs. With this belief, I established my own lab at Peking University in 2020. I tend to study plant phytochrome and have made several progresses. Let me introduce now. Light is a crucial environmental signal that regulates the entire life cycle of plants. Currently studied plant receptors mainly include the UVB receptor UVR8, blue light receptor such as photo and cries, and red or far-red light receptor phytochrome, as also PHY for short. Our lab mainly focus on phytochrome. The canonical function of phytochrome is to act as a morphogenic switch. After seed germination, plants initiate a dark green program in the absence of light, characterized by hypocotyl elongation, closed cotyledon, and etiolation as shown in the movie. However, once seedlings emerge into the light, plants activate a light green program featuring inhibition of hypocotyl elongation, leaf development, and de-etiolation. These dramatic developmental transitions are mediated by the phytochrome signaling pathway. In brief, the dark state PR form of phytochrome undergoes photoconversion to the active PFR form. PFR then translocates from the cytosol to the nucleus. Then it binds and inhibits PF transcription factors, thereby facilitating the morphogenic switch. The mutant phenotypes of PHY and PF validate their functions. The PHY PF signaling modules have been studied for nearly 3 decades, yet its underlying molecular basis remain unclear. Moreover, despite PHY and PHY-B being the most extensively studied phytochromes, the molecular basis for their divergent signaling pathways is poorly understood. However, PHY and PHY-B share 52% sequence homology and nearly identical domain architectures. It's hard to figure out their functional divergences according to the sequence. Then we wonder whether the structural approach could be informative. In 2022, Professor Wistra, pioneer in phytochrome structure biology, reported the first full-length structure of PHY-B in its dark or PR state. These breakthroughs enables us to stand on the shoulders of giants for further phytochrome research. To purify phytochrome proteins, we should first know about its biochemical and spectral properties. Phytochrome consists of a multi-domain protein and a billing molecule named P5B. The PR state exhibits a red light absorption peak at about 660 nanometers, while PFR shifts the peak to about 720 nanometers, indicating a far red light absorption. Therefore, we developed a novel purification strategy for plant phytochromes using bacterial and insect cell systems, which is soon be published. Then we try to figure out why phi A is more sensitive to light than phi B. We know phi A functions during the dark to light transition of seedlings, but phi B dominates the light growth of adult plants. Consequently, phi A exhibits about 1,000-fold higher sensitivity to weak light than phi B. With phi B PR structure solved in 2022, we continued to determine the structure of phi A PR. Phi A PR is also a dimer, and its N-terminal PSM and the middle hinge ranging PES2 domain form a plane, and the C-terminal domains protrude above the PSM PES2 plane. Now, let's see the structural differences between phi A PR and phi B PR. We can see the PSM PES plane blue in phi A. Then we show phi B in gray. The two paths align well. However, the HKRD, the C-terminal HKRD domains, green in phi A, dark gray in phi B, largely diverge. Phi B's HKRD bends back toward the N-terminal plane, creating strong intramolecular interactions. This explains phi A's higher photosensitivity. We think so. Its PR state is less stable due to weaker interactions between C-terminal HKRD and N-terminal plane, facilitating easier photoconversion. Spectral data from Richard group and ours support this model. We can see full-length phi B requires intense light for activation, but phi B lacking the HKRD like full-length phi A respond to weak light. We also solved the structure of maize phi A, and it mirrors Arabidopsis phi A, indicating the evolutionary conservation of this mechanism. In summary, we uncovered the structural basis for phi A and phi B light sensitivity divergency. Next, we aim to solve PFR structures to unravel the molecular mechanism of phytochrome photoactivation. We obtained higher quality protein samples of phi B in both PR and PFR states. However, due to the intrinsic flexibility of phi B PFR, we were unable to solve its standard alone structure. As an alternative, we reconstituted a complex of phi B PFR bound to the PIF segment and successfully determined the complex structure. We can see although the full-length 5B proteins were used, only the N-terminal PSM engaged in PIF6 binding, as shown in the movie. The PSM dimer, as shown by the arrows, transitioned from a head-to-tail arrangement in PR state to a head-to-head conformation in PFR state. In addition, the C-terminal PAS2 and HKRD region in PR state becomes absolutely flexible in PFR. Furthermore, intriguingly, the 5B PFR dimer binds only one PIF6. Now, let's see how 5B transduces red light signals into conformational changes. First, red light absorption by P5B molecule triggers 180 degrees flip of its the fourth ring, also the D ring, and followed by its sliding. Then these changes from P5B propagate to a tan-like structure in the following phi domain, and the phi tan undergoes transition from beta sheet to alpha helix. To prove the importance of the alpha helical tan in PFR, we can see all the three mutations to serine 584 abolished the 5B photoactivation and compromised the 5B function in arabidopsis thiolinase. In PR state, the bit sheet phi tan, the green one, is close to a loop region called mode loop, the yellow one, in the following PAS2 domain. But when the phi tan is transformed into alpha helix, the pink one, it sterically clashes with the mode loop, thereby disrupting PAS2 mediated interaction and that stabilize the 5B PR dimer. Our biochemical data supports this structure observation. We can see the 5B 908 segment containing the PSM PAS2 region exists as a dimer in PR, but shifts to monomers in PFR. Due to the weakened interactions between the N and C terminal region of 5B, the PFR becomes highly flexible. And early work from Corey's lab revealed that interactions between the N and C terminal regions of 5B becomes attenuated in PFR state in accordance with our structure observation. Now, let's see how 5B PFR binds PIF6. We can see PIF6 segment containing the EPB motif folds into two parts. N-terminal beta hairpin called EPB beta mainly binds to one 5B PSM monomer, and a C-terminal alpha helix called EPB alpha binds to the dimerization surface of two 5B PSM dimer. This 5B PIF binding mechanism is supported by previous studies. An early study from Quill's lab revealed that PIF APB is sufficient for 5B PFR binding, with PIF6 APB showing the strongest affinity. And a genetic study from Nagatani's lab proved the nuclear localized PSM alone complements 5B loss of function as shown by the purple dashed line, while dimer enhanced PSM with gas mimics a 5B oral expression as shown by the red dashed line. We further looked into all the key residues involved in 5B PIF6 interaction and confirmed their roles by biochemical and genetic data. Then let's see why the 5B PFR dimer only binds one PIF6 APB. By comparison of the two 5B PFR monomers, we can find two asymmetric parts, and both are involved in PIF6 binding. So the structural asymmetry of a 5B PFR dimer would create steric hindrance to one more PIF6 binding. And our biochemical data further support this observation. Here we proposed a model for PIF6 binding by 5B PFR. PIF6 APB stabilizes the flexible 5B PFR by anchoring the N-terminal beta hairpin to the NTE domain of one 5B monomer and bridging the C-terminal F helix across the head-to-head dimer interface. We also solved the structure of a PIF6 bound 5B mutant with tyrosine 266 to histidine. This 5B mutant identified by Lagrius lab is constitutively active and exhibits light green phenotypes in the darkness. It's near identical conformation to 5B PFR PIF6 confirms all our previous conclusions. So through this work, we uncovered new details about 5B gets activated by light and then binds to its PIF partner. But of course, there's still more to discover. For example, how exactly 5B gets into nucleus and how 5A specifically interact with PIF. This will be the focus of my future research. We will discuss now. We have learned that 5B carries an intrinsic nuclear localization signal and enters the nucleus autonomously upon activation. But 5A requires the shuttle factor FHI1 to nuclear import for nuclear import. We have already successfully prepared the FHY1 segment bond 5A PFR protein complex and are now working to solve this structure. This should reveal exactly how 5A's nuclear entry works at the molecular level. Then let's talk about the PIF binding specificity. All 6 PIF members in Arabidopsis combined 5B PFR through the APB motif, but only PIF 1 and 3 have a special APA motif needed to bind 5A PFR. Similarly, we have already obtained the PIF APA segment bond 5A PFR protein complex and are currently working to determine its structure. Furthermore, when PFR binds to PIFs, it triggers rapid phosphorylation, ubiquitination, and degradation of PIFs. And recent work from Chen group added another layer. They showed PIF binding could directly block PIF's transcriptional activity. Our own biochemical data further indicate the activated 5B physically prevent PIFs from binding to DNA. But how exactly these regulations work at molecular level is still an open question. Finally, I want to talk about how our work matters for agriculture. It's about seed avoidance. When plants sense seed through reduced red or far-red light ratios, inactivated 5B triggers them to stretch their stems, early flowering, and low biomass. is better for close planting and productivity. Our ongoing solution is to engineer 5A and 5B's photosensitivity. Then we can potentially design shade tolerant crops. We are already planning to test this approach in maize, and if successful, this could revolutionize how we grow crops in high-density plantings. Finally, I express my deepest gratitude to my advisor, Professor Jie Chai, and collaborators, and all my lab members. Thank you.
Thank you very much, Dr. Wang. Any questions from the committee member? Thomas, please. Thomas, please.
Oh, I have no question. I was just applauding. But I think Christian has a question.
Hello, many thanks for this great talk.
Hello.
There is some work, I would say mostly from the group of Eberhard Schaefer, which proposes, and I think the data is actually quite good, but a bit indirect, that one reason why Phi-A is more sensitive than Phi-B is because Phi-A can work as a heterodimer, one being PR and the other one being PFR, while all the other phytochromes, they need to be PFR homodimers. And so I was wondering whether you think that such a Phi-A PR-PFR heterodimer, you would be able to see one of those using the structural approaches that you are so nicely using.
Okay. Thank you for the good question. In fact, we have not got a structure of a heterodimer of PR and PFR heterodimer. We have purified phytochromes from different species and process these proteins by different conditions of red light irradiation. And we solved all these structures, but it's very strange that we never got the PR and PFR heterodimer, and I think this state is probably a intermediate state, and it's very unstable maybe in the solution. And we use the cryo-EM to solve the structures, and it requires the protein samples to be very stable. So I think maybe other approaches in structural biology should be used to solve the PR and the PFR structure.
Thank you very much. Can I ask a follow-up question or somebody else?
Yeah, sure.
So PIF6, as you showed us, is very special because it binds phytochrome B apparently much better than the other PIFs.
Yeah.
Do
Yes.
you think that it also has a somewhat different mode of interaction with phytochrome B than, I don't know, the PIFs like, I don't know, PIF3, PIF1, PIF4, for which we actually also know what they do physiologically with mutants, because PIF6, its role is a bit mysterious.
Yeah, again, thank you for the very inspiring question. In fact, we, as shown in the pull-down acids, and we get 4 different PIF and 5B PFR complex. And we also have tried to solve all the structures, but only the PIF6 and 5B complex was solved. The structure as shown in this. And I want to see the residues in the APB alpha helix part is not very conserved between these PIFs. And I thought maybe this part determines more probably more modes of 5B PIF complex.
Thank you very much.
Thank you. Thomas, would you like to ask one more question?
I don't have a question. Thank you.
Okay, thank you. Okay, let's move on to our next speaker, Dr. Xiaobo Li from Westlake University. Today he will share his journey to enhance the photosynthetic energy capture. Dr. Li, please.
Okay, can you see my screen?
Yes.
Okay. Thanks, Lucian, for the introduction. Hi, everyone. I would like to first thank Molecular Plant for this great opportunity for me to participate in this event, and also the opportunity to present to you our work. My lab works on photosynthesis. Photosynthesis is the process that supports human civilization by providing various resources and powers the entire biosphere. And the step in photosynthesis that we are most interested in is actually the very first step, the harvesting of light energy, which is performed by the complex known as light harvesting complexes. In such a complex, there are structural proteins known as LHC protein, and also chlorophyll and carotenoid pigments. These two kinds of pigments harvest the photon energy, transfer the photon energy to the reaction center of a photosystem supercomplex to enable chemical energy generation. This is the first step, and it's also a step where energy is lost very prominently. The crops that we care most about and green algae belong to the so-called green plants. Green plants have chlorophyll A and chlorophyll B and some carotenoids in their LHC. And these pigments harvest blue light and red light only, but not much green, yellow, or far red light. Consequently, more than half of the solar energy actually cannot get absorbed by the complexes. And what makes the things, and this is under normal conditions, what makes the things worse is that under stress conditions, the energy loss is even more obvious. Under some stresses, plants get chlorotic, that means chlorophylls are getting lower. There is less absorbance, and under some other conditions like excessive light, some energy is lost as heat, and there could be damages in the photosynthetic apparatus, leading to even lower photosynthetic energy intake. And my lab is working in both issues. In the first area, we dissect pigment biosynthesis pathways from algae that are not green. They have pigments that may complement pigments in plants in light absorption. In this way, in the end, we hope to let plants get more energy. In the other direction, we dissect regulatory network of photosynthesis under various stress conditions, and I hope it's in the end, rewire the regulatory network. So photosynthesis is de-repressed. It's sustained under stress. And in today's talk, I will only discuss our efforts in the first direction regarding the light spectrum. I will first tell you about our progress in the past several years in dissecting the biosynthesis of pigments in diatoms, a large group of algae, and these pigments enable harvesting of green light. And I will then spend several minutes on our preliminary future plans on combining systems and synthetic approaches to broaden the light harvesting spectrum of green plants. For the first part, between the two kinds of pigments, carotenoids are relatively diverse, but there are only a small number of chlorophyll molecules that are common. For oxygenic phototrophs, there are in total 5 chlorophyll species, known as chlorophyll A, B, C, D, and F. And almost every organism has chlorophyll A, so it's essential. And the other 4 are typically accessory. They help chlorophyll A in light absorption. They absorb at different wavelengths. And the one that we worked the most on is chlorophyll C. It actually has several structural uniquenesses compared to all the other 4 chlorophylls. For example, on the microcycle ring, there is a single bond in chlorophyll A and the others, but a double bond in chlorophyll C. And on the side chain here, it's also a single bond in chlorophyll A, but a double bond in chlorophyll C. And most prominently, all the other chlorophylls have a long alcohol chain, the phytyl chain, esterified to the carboxyl group. But for chlorophyll C, it's a free carboxyl group. So it's more hydrophilic and it's smaller. The fact that different groups of photosynthetic organisms have different chlorophylls was known actually a long time ago. It was found by this Irish scientist, George Stokes. In his 1864 paper, he first pointed out that plants have 2 kinds of green substances. Previously, people thought it's just one. And then he mentioned that he found that in olive-colored seaweeds, the second green substance is replaced by a third green substance. Using today's terminology, the plants and green algae use chlorophyll A plus B for photosynthesis. And those olive-colored groups, which are actually the main primary producers in the oceans, use chlorophyll A plus C for photosynthesis. You see that they all look olive-colored or brown-colored because they actually have a higher amount of carotenoids. And the feature of chlorophyll C at least partly contributes to the high carotenoid content. Here, we are looking at the structures of the monomer, LHC monomer in a green plant and in a diatom. We see that chlorophyll A here has a tail, and in a conserved site, the chlorophyll C has no tail, leaving space for an extra molecule, and in this case, it's xanthine. So then if we compare the major pigments between diatoms and green plants, the chlorophyll C has a high peak in the blue light region, and fucoxanthin is a special carotenoid. Compared to the common ones in plants, its absorption is redshifted towards the green region. So in total, diatoms would absorb more light in the blue-green region, and which penetrates the best in water. So this is definitely a good strategy for their survival in the marine environments. At the same time, because of the complementarity between these pigments and plant pigments in absorption, they could have translational potential in plant synthetic biology. However, their biosynthetic paths long remain elusive, so that limits the utilization of these pigments. And one reason for that is that for all the chlorophyll C containing algae, the genetic approaches have been difficult. This is a model diatom called PT, Phaeodactylum tricornitum. It has been transformable since last century. However, if we want to knock out a gene, it was difficult because it's a diploid. And unlike Arabidopsis, it has no sex cycle. We cannot do cross. So using old approaches, we cannot easily generate homozygous mutants, which should have a phenotype. But recently, colleagues in the community developed CRISPR-Cas9 procedures in this organism. And Huan Zhang in my lab improved the procedures. And now we have our own protocol for PT transformation. We put Cas9 and guide RNA gene on a plasmid and put the plasmid in E. coli. Interestingly, although diatoms are eukaryotic algae, they are compatible to conjugation. By mixing the 2 organisms, the plasmid can get into the nucleus of this diatom, and it can, if Cas9 works efficiently, we should get bioallytic mutants. With this approach, now we think about what kind of genes could be important for chlorophyll C biosynthesis. Chlorophyll C is actually pretty similar to DVP in structure. DVP is a common precursor. It's required for also chlorophyll A and chlorophyll B. So it seems that if a single bond here in DVP is converted into a double bond, chlorophyll C is actually easily generated. So we just need one enzyme, actually, an enzyme that can convert a single bond to a double bond. However, cells have many kinds of oxidative reductases. Actually, many of them can have the ability to enable single bond to double bond conversion. They require different factors. Some of them require oxygen, some of them do not. So if we consider all of them, there are hundreds of candidates to try. Yanyu and Tianjun in my lab responsible for this project, they prioritize the candidate genes based on co-expression with upstream genes in chlorophyll biosynthesis. We also look at predicted localization, and the enzyme should be in the chloroplast stroma. Diatoms have a complex plastid chloroplast with 4 membranes, but still there are softwares that can predict localization to some degrees. Besides these biological considerations, my colleague Li Han in the chemistry department also suggested us to look at the local chemical environment of this reaction, because that can affect how active the atoms are. In literature, there is an antibiotic synthesis. In this reaction, it's highly similar to the reaction in chlorophyll C. In both cases, the double bond is formed on the alpha carbon and beta carbon atoms of a carboxyl group. And this reaction is catalyzed by a dioxygenase. So in our list, we also prioritized dioxygenase genes. And after knocking all of them out, we found that for one of the genes, we immediately saw clones, transforming clones with different colors. The brown one is normal normal diatom color, but the green one is actually mutated color. It's abnormal. We sequenced the different clones in the guide RNA region. We found that all the brown ones are wild type, and all the green ones have either frame shift mutations or large deletions. And we also had complementation evidence later. So we know that disruption of this gene leads to green coloration. We then did HPLC to analyze the pigment composition of the mutant. Compared to wild type, the mutant completely lacks the 2 chlorophyll C species, but instead accumulates their corresponding precursors, DVP and MVP. And this does look consistent with what we expect for a chlorophyll C synthesis mutant. Interestingly, in parallel, if we look at the carotenoid fucoxanthin, the ratio of carotenoid to chlorophyll A is also lower in the mutant. This explains why the mutant looks greener. It loses chlorophyll, but it also loses the green light absorbing carotenoid. That's why it looks greenish. And these 2 pigments, chlorophyll C and fucoxanthin, they could be codependent on each other for stable accumulation. At least one reason is that they are adjacent to each other in the light harvesting complex. But of course, it's also possible that the entire complex is degraded. So then to test whether this enzyme is the chlorophyll C synthase, we purified the precursors from the mutant and made recombinant proteins in E. coli. By mixing them together, we easily got chlorophyll C. And when the requirements for this enzymatic reaction are the same with what's required for other dioxygenases. So it's a typical dioxygenase, but this gene is our novelty is the finding of this gene that people have long sought after. In nature, there are many groups of algae that use chlorophyll C, including diatoms and also haptophytes, which actually precipitate calcium carbonate because they make calcium carbonate in their shells. And also dinoflagellates. They are important for coral formation. They also use chlorophyll C. They all have homologs of diatom CHLC, and we prove by testing the activity of them, we prove that they are functional. Interestingly, there are also exceptions. There are some multicellular algae known as brown algae, including the edible kelps. They also have chlorophyll C, but they don't have CHLC actually. This algae, including these 2 and also haptophytes, dinoflagellates, they emerged after complex endosymbiotic events. It's believed that there was a single primary endosymbiosis event with the cyanobacterium engulfed, leading to the chloroplast in green plants and red algae. And that's a 2-membrane chloroplast. And chlorophyll B likely emerged in the green lineage as a new invention of that lineage. And red algae only have chlorophyll A. Then in secondary endosymbiosis events, the red algae got engulfed. It became the complex chloroplast in those algae. Because there was it's believed that there may be common origins for diatoms and kelps. So people thought that chlorophyll C may be from that common origin, a common ancestor at a certain stage, but clearly calves do not have any homolog of CHLC. So they must have evolved the chlorophyll C synthesis ability in a separate event. And this is distinct from previous hypotheses. It is also distinct from chlorophyll B, because the chlorophyll B synthesis enzymes are all homologous to each other. This is our chlorophyll C synthesis story. And I mentioned to you that in diatoms, chlorophyll C is often together with fucoxanthin. And fucoxanthin is actually the main contributor of green light harvesting. We are also interested in its biosynthetic path. In this project, we had on a conference, I learned that Professor Martin Law and Professor Graham Pierce had overlapping interests. We found that our data are actually complementary to each other to some degree. So in the end, we decided to collaborate, and we published the early steps, the upstream steps, and the final step of fucoxanthin biosynthesis. Here, I show the pathway from a common precursor, neoxanthin, which is also present in plants. So if we want to make fucoxanthin in plants, we needed the next 5 steps. And we have found 3 of them. The mutants lack fucoxanthin, and there are 2 enzymes that are remaining to be found. So this is our work in the pigment biosynthesis area in diatoms. In the next part, I will share with you research plans on identifying more pigment biosynthesis enzymes, making more pigments, and also engineering green plants. This is a very preliminary, actually. So today I'm looking forward to your criticism and feedback. First, we wanted more pigment choices, not just for green light, but also far red light and yellow-green light. For green light, our direction is obviously to finish the fucoxanthin biosynthesis pathway. And far red is very interesting because it's actually enriched in the middle or lower canopy to a higher degree than green light. And in nature, there are 2 chlorophylls that can harvest the far red light, chlorophyll D and chlorophyll F. They are both from specific cyanobacterial groups. And compared to chlorophyll A in the red light region, they are red-shifted, so they can absorb around 700 nanometers in vitro. And in vivo, there may be, in some places, there is a further red shift, so they could absorb up to 730 or 750 nanometers. So they also have interesting translational potential. Between those 2, for chlorophyll F, the synthesis was found several years ago. But chlorophyll F synthesis is actually distinct from most other enzymes. It's actually a large complex. It's basically a mutated photosystem 2. And if when people move some of the subunits to other cyanobacteria, they can lead to chlorophyll F, but those subunits have to be assembled into the large complex. So far, there is no chlorophyll F production in eukaryotes. It may be hard to make such a complex in eukaryotes. For chlorophyll D, the synthesis is not known yet. And there are many different cyanobacteria that can make chlorophyll D, but in 1999, it was found that this cyanobacterium actually makes chlorophyll D as its main pigment. More than 90% of the chlorophylls, actually much more than chlorophyll A. So we believe that it has a potent chlorophyll D synthesis. We recently started growing this cyanobacterium, and we hope to establish genetic methods in it to enable candidate gene knockout. And so that's the green light and far red light part. And although the chlorophylls and carotenoids differ from each other in absorption, none of them can easily cover the green-yellow region. If we can have a pigment that absorbs in this region, it could be more complementary to plant pigments. What we want to do is to design artificial pigments. If we synthesize a random pigment, it's not going to work because it can not transfer energy to other pigments. It cannot be bound by LHC. So we want artificial chlorophylls or carotenoids. For example, we have 2 carotenoids, fucoxanthin and pyridinine, and they have chemical groups modifications that lead to a red shift in their absorption spectrum. And we could design a carotenoid that combined the features that could be an even further redshift. By making such a structure, my colleague, Professor Jing Huang, a quantum chemist, predicts that this chemical could absorb in the yellow-green region. And because these groups are from the natural carotenoids, we may be able to get it compatible with biosynthesis. We just need to figure out which enzymes are important for making the groups in those two pigments, and hopefully we can modify their substrate specificities and stack the reactions on the same precursor to lead to such a molecule. So these are the pigments part. We thought about green light, far red light, and yellow-green light. And then the more difficult part is to design to get the pigments into functional photosystems. So we thought about two strategies at this early point. In both cases, we would leave the chassis, green plant chassis reaction center unchanged. And then we need foreign pigments for sure. Then there are two choices in LHC. We could move the foreign LHC together. So we generated the entire light harvesting complex in green plant. Then we need to engineer the interface between LHC and a reaction center. For the other strategy, we can use the green plant's own LHC. In this way, it may need to be engineered to be able to harbor the foreign pigments. For either case, it will take a lot of a huge amount of trial and error, and we think it's not realistic to do it in a land plant at this moment. But green algae are close to land plants, and they are quite conserved with each other in photosynthesis. So we are considering using green algae as a test bed. For example, this unicellular green algae, Chlamydomonas ringharti, is amenable to genetic manipulation. And we will need high throughput approaches if we need to generate different versions of the LHC proteins. Then, so we need large scale screen capacity. At this moment, we have established a high throughput analysis of pigment phenotype and chlorophyll fluorescence phenotype in Chlamydomonas ringharti. And besides chlamyd, we recently started growing another green algae called oxynochlorella. And this one, based on some papers, it's amenable to a homologous recombination-based gene targeting. And this is certainly a useful approach in synthetic biology. We are still in an early stage in optimizing its transformation. Okay, I want to end my talk here and thank people in my lab that make the work possible. I also want to thank my collaborators in China and abroad, and thank the funding agencies for the support, and thank Zhejiang University and Fudan University for the joint PhD program. And I want to stop here for questions. Thank you.
Thank you. Thank you very much, Dr. Li. Any questions from the comment member? Please just feel free to turn on your microphone. Ralph, please.
Hi, Ralph.
Hi. Well, thank you very much. That was really nice. I think the design of artificial pigments is a very interesting concept, but one of the challenges that comes with this is that the substrates, like the carotenoids, are highly hydrophobic molecules. So this is not easily amenable to in vitro evolution approaches to generate the required enzymes. So what would be the strategies? How do you want to go about evolving the suitable enzymes to make these new chemistries?
So I guess in the end, we could do the old style directed evolution just by making random mutations. And but before that, we would like to screen different organisms. We found that although many organisms share the same biosynthetic step of fucoxanthin, they have different substrate specificity. Some of them are relatively strict. Some of them could be loose because they don't have the other substrates. There is no evolutionary selection pressure against the other substrates. So I think the first step is actually to screen different organisms for a loose substrate specificity.
Mm-hmm. And then just for my own understanding, because it wasn't entirely clear to me why you need the bacterial conjugation to do the CRISPR-Cas in Pheodactylum. What's the advantage of the conjugation strategy over like conventional biolistics or other methods?
Actually, the short answer is just for us, it gives us more clones. Yeah, biolystic is the classic method for this organism. And for some groups, they can do electroporation. But interestingly, the electroporation is hard to reproduce. Different groups have very different efficiencies. And for the bacterial conjugation, we don't need any special equipment. We just need, for example, 10 milliliters of culture. We can get 1,000 clones. Actually, that's a pretty efficient. But of course, that just means the plasmid is in. For whether it's knocked out or not, it depends on the gene. For this gene, we happen to be lucky. We got 50% of the clones to be knocked out. But for many other genes, it's much lower.
Do you have to remove or modify the cell wall to get the conjugation to work efficiently?
Actually, no, not for this diatom. Pheodactylum is many people think it's not a representative diatom, but I think it's representative in photosynthesis, but it actually has a relatively thin wall. On the SEM, very often we don't see the silica cell wall. So this one happens to be easy to transform.
Okay, thank you.
Yeah.
Thomas, please.
So a very simple question from a non-expert. So I'm wondering, you mentioned that cyanobacteria or certain cyanobacteria are producing chlorophyll D and F, which are absorbing light in the very far red light. So I'm wondering, what is the biological advantage of being able to absorb light of low energy that you cannot transfer maybe to other systems?
Yeah, that's a good question. Actually, these organisms typically live on the other organisms. So the simple explanation is that above them, there are some other algae that have absorbed a lot of the visible light. This has been shown. Yeah, they often live underneath of some other organisms.
Okay, thank you.
Yeah.
Chris, please.
Thank you very much. I was wondering if you considered using experimental evolution with Chlamydomonas, growing them in green light for I don't know how many generations, and see whether that will tune the sort of light harvesting pigments to improve photosynthetic activity, which would be kind of a complementary approach to the ones that you have proposed.
That's a great question. We are considering Chlamydomonas for engineering to test these concepts. Actually, one reason is that it may be amenable to evolution in the lab. For example, Chlamydomonas can grow in the presence of acetate. So if we disrupt its photosynthesis, we'll give it acetate, it can grow, but then we can lower and lower the acetate and see how it adapts. This is certainly useful in engineering the complexes. But for the pigments, my naive understanding is that if there is no new enzyme, maybe it's not easy to generate a new pigment, then it cannot absorb green light. But if then I'm not sure how fast that a totally new enzyme would evolve to work on those. But it's an interesting thought.
Thank you.
Okay. If there's no more questions from the community, thank you again, Dr. Li.
Okay,
And
thank you.
we can move on to our last speaker for today, Dr. Yanran Li from University of California. Today, she will share her story about how to harness microbial cell factories to uncover the essential stragolactones. Dr. Li, please.
Great, thank you. Can you hear me?
Yes.
Perfect. So first of all, I would like to thank Molecular Plants for providing us this wonderful opportunity to share our recent work efforts on harnessing microbial cell factories to uncover stragolactone of its essential biological functions. I think with this group of audience, I do not need to persuade you how amazing and wonderful plants are. So as we all agree, plants are sessile organisms, which means that once they germinate, they stay here forever. And because of that, through thousands of millions of years of evolution, plants has evolved a very rich and outstanding, wonderful secondary metabolism to communicate and interact with the environment. And what we are really interested in is a plant secondary metabolism. As a synthetic biologist, our expertise is resides in building up the microbial cell factories and developing tools. And by using these biological tools, we think we will be able to providing better tools to study how and why plants produce all these molecules, these biological bioactive molecules. So this is a landscape of what my research group is recently focusing on, basically two parts. And today, what we will mainly focus is on the left panel and specifically zoom in into stragolactone. So before I go into more stories that we have done, let's first see what is stragolactone. To give you a brief, I'm sure you know stragolactone, but just a little bit intro on the chemistry part. So stragolactone was first discovered in 1966. The first one was discovered as strigol, a structure shown here. At that time, it was discovered as a signaling molecule from cotton to induce parasitic wheat seed germination. So let's see the stragolactone structure. As you can see, it contains this tricyclic ring with this D ring. And normally most stragolactone contains this structure, and that's where we call stragolactones containing this structure as canonical stragolactone. And the ones, of course, the stragolactone that doesn't have this kind of typical triplet ring plus single ring structure is called non-canonical stragolactone. Within the canonical stragolactones, depending on the stereochemistry of serine, we further classify them as S-type and O-type. Later on in 2005, stragolactone was found to play an essential role in stimulating avascular mycorrhizal fungi, making it an even more important signaling molecule. And in 2008, it was proposed to be not only a signaling molecule, but a phytohormone that involved in many events in plant development, growth and development, specifically, most importantly, branching inhibition. Through many years of study, we kind of developed this hypothesis that canonical stragolactone is more likely to be involved or playing a more important role in the interorganismal signaling, while non-canonical stragolactone are mainly function as hormones. Here is a summary of most of the stragolactone. There are still some that's newly discovered that's not updated here, but these are kind of the majority of stragolactone that has been discovered to date. And as you can see, there are a lot. One thing that I want to point out is we want to understand that most of the stragolactones were primarily detected, isolated, and discovered from root or root extrudes. There are many questions. Although we have discovered so many stragolactones, there are many questions still remains to be answered. Just to list a few here, like we don't know why and how plants evolved to produce such diverse stragolactone structures. We don't know what the role of each of these different stragolactone structures are in different plants. We don't even know what is a real branching inhibitor and what is a real EMF inducer. And the reason that all this question has not been answered, one thing is because stragolactone is a newly discovered group of plant hormone, but the other thing is also like we don't have access to all these stragolactones. Same as many other phytohormones, and specifically in the case of stragolactone, they normally exist extremely low abundance in plants, normally nano to picomolar range. Taking another compound that we study in our lab as example, brasinosterol. Brasinosterol structure was uncovered through NMR analysis of 10 milligram. That's purified and isolated from 230 kilogram of pollen. And stragolactone is even worse. This is the photo provided by my collaborator, Professor Xiaonan Xi. And this is a setup of how we extract the xylem from poplar. And through a calculation, at least 340 liter 340 liters of xylem sap is needed to get barely enough of NMR, which means 0.1 to 0.01, 0.01 to 0.1 milligram. And that doesn't count all the losses during isolation and purification. And even worse, stragolactone is not stable, as you can see from this structure. So for many years, stragolactone community has been using the synthetic analog called GR24 to study all the related biological acids to uncover the function of stragolactone. And of course, you can imagine by using the synthetic analog, it's no way for us to clearly identify the so-called structure-function correlation. As a synthetic biologist, we ask if we can building up the microbial cell factory to produce different kinds of stragolactone and help our colleagues to solve the problem. So before that, let's look into how stragolactone is synthesized so that we can building up the microbial cell factory. Stragolactone is apocarotenoids that synthesized from all trans beta carotene with 3 enzymes working in chloroplast to convert it into carlactone, which is a key branch inhibitor. Then CYP711, which is also called MAX1, the cytochrome P450 will convert carlactone into carlactonic acid, where it oxidizing this carbon-19 to this carboxyl group. Then some more cytochrome P450 will function to convert CLA into canonical stragolactone, and other enzymes to convert CLA into non-canonical stragolactones. A side effect, when I first joined Riverside, decided to start to working on apocarotenoid, my wonderful collaborator and mentor on stragolactone project, Professor Dave Nelson, was already a well-known expert in stragolactone. So I asked him, hey Dave, do you think I can get any of this compound called carlactone so that I can utilize as a standard? Just a little is fine. So Dave reached out to all his collaborators around the world and gave me the answer no. Reason for that is because this is so unstable, and it's no way for them to ship to us from overseas. Just a little fun fact for you to understand how unstable stragolactones are. However, this biosynthetic pathway, because it's compartmentalized between plastid and ER, is basically impossible for us at that time to building up this whole biosynthetic pathway either in yeast alone or in E. coli alone. So what we did is we utilized the consortium mimicking what's happening in plant cells, using E. coli to mimic what's happening in plastid and yeast to mimic what happens in ER. And by using this microbial consortium, we was able to synthesize an array of stragolactone. But just to be noted that the titer is really low at that time, is 5 to 15 microgram per liter. But hey, we are able to produce all these molecules. They are detectable using LC-MS. And we have a lot of standards at that time. So we were like, we will start using this microbial consortium, this biosynthetic tool, a synthetic biology tool, to see what questions we can answer. So by looking at the biosynthesis of stragolactone, what we find is a little bit intro into the background. We find that carlactone and CLA are the key branch inhibitor, and starting from here, we are going to produce this canonical stragolactone. We are really interested in how canonical stragolactone are produced because they are very important interorganismal signaling molecules. At the very beginning, we don't know how canonical stragolactone was synthesized until 2014, the first paper published on characterization of 2 MAX1 variants from rice, encoded by rice, that's able to convert CLA into urbanco. So for a long time, people have been believing that MAX1 analog variants are the one that's actually converting, not only having the conserved function, converting CL to CLA, but also converting CLA to canonical stragolactone. Then around 2019, this group of enzyme called CYP722C was uncovered to be able to also catalyze the conversion between CLA into either 5-DS or urbanco. So at this point, we don't know which one plays a major role across flowering plants. We do find that CYP722C exists in generally exist in flowering plants, and we want to ask, are they functionally conserved? We also see many plants encode more than one copy of MAX1. We don't know what their functions are, so we want to also characterize their function. And behind that, we want to answer the question, which one is a major contributor to canonical stragolactone structures? And we also want to ask how and where plant gain the canonical strag-lactone synthetic capacity. So first of all, let's look into those max van variants. What we find is many plants in rose family and the grass family actually encode at least 3 max1 analogs. Just a reminder, the first canonical strag-lactone synthase, the max1 variance is here on the phylogenetic analysis. What we did, but we don't know the function of all the other max1 variants from all these species. So we just synthesized all of them, throw them into the consortium, and identify what compounds they are producing. And this is the results. Turn out most of them are either non-detectable or produce CLA. The one that doesn't produce CLA, produce something new, is highlighted over here. And among all of them, the only one that's related to canonical strag-lactone are the one from peach and the one from sorghum. The one from peach is very amazing. I just want to quickly point out that it's one enzyme catalyzing 4 oxidative steps at 2 different carbons, and that's confirmed through oxygen 18 fitting. And we also want to quickly point out that for peach, specifically this plant, in addition to encoding a ppmax1 variants called ppmax1c that converting CLA into striga, it also encodes a CYP722C, which is conserved across all the dicots. So, and specifically in peach, this pp722C is can convert CLA into Arbanco, which is the O type strag-lactone. So I want to point, we will come back to this slide later on, but I just want to point out this very interesting fact that peach evolved really hard diversified max1 to produce, in addition to CYP722C, to produce both type of strag-lactone. For sorghum, we found one max1 that producing 18-hydroxy-CLA that's believed to be the precursor of strag-lactone, of canonical strag-lactone, actually. And what's interesting over here that we want to point out for this discovery is at that time there is a very nice pioneering study on sorghum, 2 mutants of sorghum. One is encodes LGS1 and they are susceptible to striga, and one doesn't have LGS1 through evolution, kind of lost this enzyme, and they become resistant to striga. LGS1 is a sulfur transferase, but at that time, people have no idea what's its function and why it is the switch for sorghum to be susceptible or resistant to striga. So one point the one thing I want to point out is the 18-hydroxy-CLA under certain acidic condition actually can be converted into Arbanco spontaneously. And we ask, now that we have this max1 variance that producing 18-hydroxy-CLA, maybe is it the substrate of LGS1? And the answer is yes. It actually, LGS1 will function on 18-hydroxy-CLA, and then with another enzyme, it'll convert into this S-type. So now with the mapping of functional mapping of all the max1 variants that showing here, clearly max1 doesn't seem to play an important role in canonical strag-lactone biosynthesis, with only peach, rice, and sorghum max1 variants being able to produce something related to canonical strag-lactone. How about CYP722C? And the answer is yes. So we were able to synthesize a whole bunch of CYP722C encoded by all kinds of different plants, especially dicots. And we found that almost all of them are able to either produce O-type or S-type canonical strag-lactone. So to answer this question, so then the answer to the question of which one is a major contributor to canonical strag-lactone become very clear. It is CYP722C. Over here, before we move on to the fourth question, I want to quickly point out one interesting observation we found. Through mapping all the CYP722C's function, we found that some plants, for example, strawberry over here, it actually encodes 2 CYP722C, and with each one producing different type of strag-lactone. So this is something I have been thinking for years. It's just I think it's very interesting to point out over here at this point. Open for discussion, I still haven't resolved, but I think it's very interesting to point out. If you look, well, most plants encodes 1 CYP711 and 1 CYP722 to produce just one type of canonical strag-lactone. These two members in the rose family, strawberry, specifically strawberry and peach, during evolution, apparently they work really hard utilizing different strategy to produce both type of canonical strag-lactone, which raises the possibility that these two type of canonical strag-lactone must have different function, and they must play a role in either strawberry and peach to adapt to different environment. And we want to know what's their function, and we are still working really hard to try to answer this question. So then we moved on to the fourth question. Now that we know which one is a major contributor to canonical strag-lactone structures, we want to know how plant gain the synthetic capacity. And by answering this question, that could be the starting point for us to gradually understand the biological function of canonical strag-lactone. By looking at the phylogenetic analysis, Dave and my postdoc Sheng at that time asked this question. Find that there is a group called CYP722A or CYP722B that's right next to CYP722C, exists broader across seed plants. What are their function? And as you probably know, the answer strategy is simple. We just synthesize a bunch and then throw them into our microbial consortium. We did see an unknown hydroxylated CLA. From the mass, it looks like it's an unknown hydroxylated CLA though. Then Anqi tested a bunch of different other variants from different plants. Turned out that it had all the CYP7 across the CYP722A and B family. This function to synthesize this unknown hydroxylated CLA is conserved. So we need to know what's its function. What's the structure of this compound? However, as you may recall, the microbial consortium's efficiency is pretty low. It's only 5 to 15 microgram per liter. So Anqi worked really hard to improve the efficiency of the microbial cell factory to milligram per liter. This is exactly the NMR tube she used with the unknown compounds inside. And by doing an MR analysis, we were able to clearly elucidate where that hydroxyl oxidation takes place on the carbon 16. So the product is a 16-hydroxyl CLA. Well, this structure is not like that exciting, but what really makes us super exciting is after we have this conversation with our collaborator, Xiaonan. What he told us by looking at the structure and knowing that compounds, compare that compounds with what he has been detecting for over the years. He told us that this is exactly the compound he has been detecting for years in various plants. What's really interesting about these plants is it present during the branching and then phase out after development. Here are two examples, pepper and pea. And as you can see, at the early stage, we do observe this compound 16-hydroxyl-CoA in pepper and pea. And at a later stage after development, we no longer detect them. And what's even more interesting is that Xiaonan was never able to detect this compound in the root, only in the shoot. And we want to point out again that, as you remember, most strag-lactone were discovered, almost all the strag-lactone were discovered and detected mainly in the roots or root extrudes. So this is actually the only strag-lactone discovered nowadays to be detected exclusively in the shoot, but never in the root. And that gave us the hypothesis, lead to the hypothesis that maybe it's related to the hormone function. We don't know. We will talk about it later. And then Dave and his wonderful PhD student, Annalise, was able to confirm that this compound is about active strag-lactone. It activates strag-lactone responses in plants. And she was then able, through delicate biochemical characterization and enzyme engineering, she was able to show that between CYP722A and C, actually, if you think about it, it's basically just a change in the oxidation site. CYP722A oxidized carbon 16, while CYP722C first oxidize carbon, activate carbon 18. And she was able to show that by only one single mutation, she is able to equip CYP722A with 18 hydroxylation catalytic capacity, which means she is able to change a CYP722A as partially as CYP722C by just one single mutation. And we think that's probably what's happening during evolution. Somehow, at certain point during evolution, one of the plant or several plants at a similar time somehow gained the single mutation, got 18-hydroxy-CoA catalytic activity, and figure out the canonical stragolactone biosynthesis and start using this compound, the canonical stragolactones, to communicate with other species. So by answering the question on where plant gain the canonical stragolactone synthetic capacity, we also want to, and before we go to the conclusion, we also want to quickly point out that during the study, what deep analysts found is that the function of CYP16-hydroxy-CoA, which is a dot, in comparison to the wild type, the positive control of GR24, it does have function, but this function is dependent on this enzyme called elbow and clamped, which means in this mutant, as you can see, they show the same phenotype as a wild type. And even more interesting, we found that the double mutant of elbow and clamped actually showed the same phenotype, same level of branching as a MAX1, 3, or D14, which are the SL deficient mutants. MAX3 doesn't produce any stragolactone, while in D14, plant cannot sense stragolactone at all. That's where they are using as a gold standard for this max branching phenotype. Well, this double mutant actually have exactly the same phenotype. So we ask, is it possible that the real branch inhibitor just be downstream of 16-hydroxy-CoA? Well, we are still starting it, and hopefully in the near future, I'm able to share a story on that. And with that, I want to show you that by using this microbial cell factory, we were able to push the forefront of stragolactone field by answering some of these questions and with more questions to be answered in the future. And hopefully I have been able also to convince you that synthetic biology is a wonderful tool. And by collaborating, we are able to together study the stragolactone metabolism, advance the forefront of this field. And by that, I would like to thank you for my students, alumni, and our wonderful collaborators and the funding sources. And thanks you all for your attendance. And I would like to answer any questions you have.
Thank you very much again, Dr. Lee. Any questions from the community members? Please, Thomas? Thomas, please.
No, it was just applause. Sorry.
Okay, thanks. If you have any questions, just feel free to turn on your microphone. Okay, Chris.
Thank you very much for this enlightening talk on the complexity of strigolactones, which I definitely didn't know about.
Thank you.
I wasn't sure I got it. What is the situation in Marcantia? Do they have the strigolactones or not? From
No.
They don't have any strigolactones.
So I think right now, like when I first started, basically all my colleagues are just using GR24. There are only one or two groups that was able to synthesize some of the, stragolactone or isolate them from plants, but really low amount. What we did, we were able to obtain the 4DO, 5DS, some of the stragolactone standards with a price of $500 for just 1 milligram. And those are racemic mixture. And as you can see, they are basically just chemical synthesized. Most of the time, they have this inventory because someone submitted a customer synthesis, and that's why they have some and they start selling. So it's really hard for us to look to find a valid resources to just get some of the standards, because we need those standards to make sure we are synthesizing those. So that's why we have those standards. Right now, after we build this microbial cell factory, our collaborator has access to many of these compounds. And we are able to, as you can see over here, to utilize a compound to do all this biological or plant assay.
But coming back to my sort of evolutionary question, when did plants acquire the ability to make strigolactones? I think it's clear that moss has strigolactone, but Markantia, I wasn't sure. And so that was actually my question. Is that, Markantia liverwort, you had a slide with the where, the different enzymes, where they are present on the genetic tree.
Oh, yeah. So,
so Markantia is on top, I guess.
Sorry, I thought you mean market. So that's
no worries.
But this is a very good question. That's what we are trying to answer. Actually, you see the 733 is a very interesting group of enzyme that we actually have no idea what their function is. But Markantia doesn't have encode even CYP733, but they do encode the enzyme required to produce CLA.
Okay.
Which means the first three enzymes to convert it to carlactone that's conserved across all land plants. And then I think it also have the max one to convert from CL to CLA. It does also have D14, which means the enzyme sensing strigolactone.
Yeah.
But and also another interesting thing, I think my collaborator has done, that's not my data though, it's they were able to find that I've got the data, exactly the data, but I think they were able to treat one of the very early land plants, and they did see some phenotype with strigolactones when treated with strigolactones. So they for sure have the signaling, they for sure can producing the early molecule. We don't know what the active form is. It could be some of the enzyme that's, for example, the clamp at elbow that further convert them into those non-canonical strigolactone that function both as signaling and a hormone. That's our hypothesis right now. But again, it's very, very early. It's just all guess. And
thank you very much.
Sure. Yeah. I just want to point out that this field is, we still have a lot of questions to be answered at this point. Yeah.
Indeed. Thank you.
Sure. Thomas, please.
So I was also surprised that there are so many strigolactones, and when I saw the list, I had the same question: why so many? So when we look at gibberellic acids, for example, we see that there are over 140 or so, but very many of them have no biological activity. So I'm wondering, do you have any biotests or something to test whether all available strigolactones are really active? Or is this, could some be intermediate metabolites or something, or are they really all, do you think they all have biological activity?
So that really depends, right? For example, in the case of 16-hydroxy-CoA, these are the biological functions. We treat them with the MAX3 mutant and to see if the branching is, inhibited. And also I think it's over here. We also work on the hydrocodone acids to see, like in different mutants, to see if they are dependent on different mutants, different enzymes. So we do have those acids, but as you can see, this 16-hydroxy-CoA in this case, for example, the branch inhibition is not amazing. The concentration we utilized is 10 times more than GR24 to be able to show the same phenotype. And that gives us a hint that it could be an intermediate rather than the final bioactive molecule. Over here, the same, as you can see. This is where the hypocodone length is in comparison to GR24. So the activity is not as strong as even the synthetic analog. So that's where we start thinking. And of course, it's dependent on elbow and clamp. And we actually also shown that elbow clamp can convert 16-hydroxy-CoA. So we know that likely it at least need to be 2 steps downstream, but we still don't know if that downstream enzyme, that downstream product is a real bioactive form. Right now, what we are trying to do is we will purify the D14, which is the receptor to sense strigolactone. And then we will conduct biochemical assay to either looking at how efficient D14 in hydrolyzing all these different strike-likeness structures, or also at the same time, the binding affinity between D14 and those different ligands to be able to find a real bioactive strike-lactone. Well, what we see is D14 should be able to recognize most of the strike-lactone, or it won't have this like phenotype change when we treat plants with different strike-lactones, but the efficiency definitely is different. And that's how
very exciting research. Thank you.
Yeah, sure.
Thank you. Any other questions? Okay. Now we have finished the first session. Thank you again. Thank you. Okay. Tomorrow we will continue with the second session of the RSPS 2025 finalist contest, where the remaining 5 finalists will share their work. We hope to see you again at the same time tomorrow. Thank you, all committee members, finalists, and all audience on the WeChat channel and YouTube for joining us today. Thank you so much.
Thank you. Good night or good morning. I don't know.
Thank you. Bye-bye.
Thank you.
See you tomorrow.
See you tomorrow.
See you tomorrow.
Bye-bye.